Thanks. That was excellent. Can I just kick off the question? I may just have not been quite concentrating enough. Where did you get your normal palmar fascia from for these studies?
That was from carpal tunnel decompression surgery. So differently to what was described in the previous study, I think.
Okay, and so that was taken as the surgeon's going into the carpal tunnel from the actual palmar fascia, or was it transverse carpal ligament? Just because we know that there are some changes in transverse carpal ligament in carpal tunnel syndrome, so it's important, obviously, not to be studying a negative pathology.
We had a discussion with this actually with the surgeons, and they confirmed that it is normal palmar fascia and not the transverse carpal ligament. Yeah.
Okay, good. And one more question just before I go to the question from Dave O'Gorman. I've wanted for a long time, and I haven't quite got around to it yet, to monitor in situ collagen turnover. So in the patient's hand before the tissue is excised, using, for example, heavy water. Have you thought about doing that?
I know that type of thing can be done. So I've seen some studies in animal models using heavy water. So not models of Dupuytren's, but in other types of work. Yeah. So yes, that's possible. I suspect it might be quite expensive, but I think it's feasible to do, methodologically feasible to do. And that would give lots of information other than the collagen turnover. You probably get information on other proteins that are turned over more quickly, like non-collagenous SLRPs proteins. So yeah.
Yeah, yeah, really absolutely.
Yeah.
So maybe we should collaborate to try and do that. Charlie.
Right.
Excellent paper, and the business of heterotrimer collagen 1 I think takes on added significance with the fact that it's relatively resistant to MMP degradation compared to the heterotrimer. And so the question I have is the mechanism. What is it that provokes? Is it just a matter of cells making more A1 strands that just randomly there's more out there, and so more heterotrimer develops, or is there any other guidance to promote assembly of one versus the other?
Yeah, that's a really great question. So potentially it can be regulated at many levels, and we've thought about this a lot. So one of the ways it could be regulated is by epigenetic regulation of the COL1A2 gene, so that COL1A2 expression is decreased. So that's been shown to be a potential mechanism in cancer because it's also present in carcinomas. It could also be the assembly process itself. So one of our PhD students has been doing some molecular dynamics modeling to see how the conditions during trimerization might affect whether the homotrimer is formed or whether the heterotrimer is formed. I think simplistically it would be nice to think it's the messenger RNA ratio, but it doesn't entirely correlate the messenger RNA ratio with whether you get homotrimer or heterotrimer. But yes, that's a good point. It's the degradation resistance of that particular collagen that
we think a cumulative thing.
Yeah, exactly.
Fascinating.
Thank you.
Yeah, it's very interesting. Dave O'Gorman wants your thoughts on the MMP versus TIMP findings, given they might counteract each other.
Yeah, so TIMPs are sort of endogenous regulators of MMPs. So therefore, if you see an increase in both, what's actually happening to the proteolytic activity? So yeah, so we've measured the production of the MMP proteins using this method, but we haven't measured their activity. So potentially, yes, the opposite could be happening. So if the TIMPs are overwhelming their MMPs, then potentially it could be producing a decrease in collagen degradation, which is what we would presume is happening in Dupuytren's disease, because the collagen's accumulating, so therefore potentially it's being degraded less. They do tend to act stoichiometrically, so sort of needing one TIMP to counteract one MMP, and the levels that we're seeing do seem similar, but it might be quite a sensitively regulated process. So that's the sort of thing we can't measure, but potentially with the deuterated water study that could be looked at.
Yeah. So again, we're not at peak data. More research needs to be done. Okay, are there any more questions at all from the floor? So I can't see any more coming in now. So in which case, I'm going to close the questioning. Liz, thank you very much. I'm going to send you back to be an attendee.
Thank you. Bye.
Thank you. And we have one final thing just before we close the session. There is a prize for the best free paper, and that's been judged by an independent panel who've been listening for the whole evening. And the results are through, and the winner of the prize, which is €100, sponsored by FIDIA, our symposium sponsors, is Sophie Reismeyer for the Association of Antihypertensive Medication with Dupuytren Disease Severity. So well done, Sophie. We'll give you a clap. It was an excellent talk. We've had 4 really excellent talks tonight, so thank you very much. And thank you to Charlie for your keynote, which kicked it all off. That was really fantastic. Please, everyone who's here, do go to our registration page. If you've enjoyed tonight, register for the rest of the symposiums. They're all going to be really good. We've got some really good speakers lined up, so please come along to those. The next one is in 2 weeks. So with that, I will end the webinar. Thank you, everyone, for attending.
Thanks.